Morning, ladies and gentlemen. My name is Weerasak Pitaksaringkarn. I come from Biotechnology, Research and Development Office, Department of Agriculture. Let me share your slide. I want to present the poster presentation. The title is Development of Screening and Detection Method of Genetically Modified Rice, LL601, LL62, and BT33 by Multiplex real-time PCR. As you know that rice is important commodity for Thailand. We have 61 million rice. It's about 10 million hectares for cultivation area and produce around 25 million tons of rice. And also Thailand is the top 3 rice exporter to all around the world. But in 2003, EU have the rapid alert system to report that the rice from Thailand has contamination of the GM. Therefore, the exporter need to send the rice sample to the laboratory for screen the GM contamination before export to another country. And also this is spend a lot of money and spend a lot of time. So this research will do the multiplex real-time PCR to reduce the cost of detection and screening tests, and also reduce the time of screening tests. By we design the 2 sets of probe and primer, as you see in the table 1. The table 1 shows the probe and primer which is used in this experiment. And the first gene is 35S parameters. We label by FAM. And the second gene, which is not terminator, label by hexalase. And the last one is the PLD gene. Is it the rice endogenous specific, labeled by sulfite. And another set is the second set. We have bar gene labeled by hexalase. We have Chi1ABAC, which is the specific to BT33 rice, labeled by FAM. And another one is LL62, labeled by hexalase. And the last one, LL601, labeled by sulfite. After that, we optimize the mixtures of PCR reaction and also the real-time PCR condition as you see in the table 2. After test many times, we found that the plasmid CRM, because in rice, it's difficult to find the CRM of the GM of these 3 even genes. So we buy the plasmid reference material from the urolfin. We found that the BT plasmid contain the Chi1ABAC and also not terminator. But the LL601 plasmid contain only LL61 even specific gene, as you see in the figure 1. And after that, we do the experiment to find the limit of detection of the LL601 plasmid and BT33 plasmid. And the last one, we also have the genomic DNA of LL62, which is provided by the urolfin. As you see in the table 3, show the limit of detection of the plasmid LL BT33, and by splicing to the non-GM rice. And the gene is not terminator, Chi1ABAC, PLD, had the limit of detection at 5 copy gene, and the rice, non-GM rice, spiked by the LL301, had the limit of detection at 5 copy gene. But the genomic DNA of LL62 had the 35S parameter, bar gene, and LL62 and also PLD had limit of detection at 10 copy gene. After that, we would like to test the efficiency of these 2 sets of primer and probe, that is it be able to screen and detect in the single spike, double spike, and also triple spike. At the lower one, you can see rice DNA spike by the single spike by plasmid and also genomic DNA, show the different CP value, and also but the noodle and also rice noodle, rice flour can also detect in different CP levels. But for qualitative detection, these two sets of primer and probe is efficiently to screening of the T5S and non-terminator, and also can detection of event specific of LL601, BT33, and also LL62. And the last table, I show that the test of 50 samples in our laboratories. We have the rice, 27 sample, noodle, 20 sample, and also flour for 3 samples. The results show that not any sample can be detected in the another GM component like 35S prompter, not symmetry, and also event specific element. Only PLD can be detected, as you see in the table 4. So the conclusion that both two sets of primer and probe can efficiently screening and detect of the event specific, and also Thailand rice still safe from the GM because the last 50 sample in our laboratories can not be detect any GM element and also event specific of the GM rice. Thank you for your attention.